So since this one has already divided, okay, it's already got a sister cell in a way over here. Okay, this was formed from one parent cell. And so whatever happened here is probably going to be fine. Okay, that one happened properly. This cell was formed properly. There's nothing wrong with this cell to begin with. Only when it's undergoing meiosis 2, something goes wrong and there will be implications on these two daughter cells. Okay, so on the left we have, non-disjunction during meiosis 1, and over here on the right we have non-disjunction during meiosis 2. So you have to think about what we're splitting apart. In meiosis 1, what we want to split, hopefully you'll see these structures are what we want to split. Okay, we want to split our homologous pairs. So we want to split one copy of chromosome 1 from the other copy of chromosome 1. One copy of chromosome 2 from the other copy of chromosome 2, so on and so on and so on. So over here on the left, okay, we are having, if you look at this top pair, this top homologous pair, that splits just fine. Okay, we have one going, let me just erase this, we have one going off to the left, and another one, this one goes off to the left, and this one will go off to the right. However, down here, with, let's call this chromosome 2, instead of splitting up like they were meant to, you'll see both homologous chromosomes are going off to the left here. Okay, so the way to think about it is what's going to happen when this cell divides. Easy way to do that, if it's drawn like this, is if you just draw a line down the middle of the cell. Right, that's what the daughter cells are going to look like. This is going to form one of the daughter cells down here, and this is going to form the other daughter cell. So this cell that will form on the right is only going to have one, well, it's going to have one chromosome 1. Let me just wipe this a bit. This one is going to have one chromosome 1, and it's not going to have any chromosome 2. Whereas this cell on the left is going to have one chromosome 1, and it's going to have two copies of chromosome 2. Okay. Two copies being, remembering that each of these copies has two chromatids. Okay, so it's got two chromosomes in the X, what we mean by like this type of chromosome with the centromere in the middle, but it's got two of those, so it's got 4 copies of that DNA inside that cell. Okay, remember that each chromatid has a full copy of that DNA. Okay, so now this cell on the right is going to be one of the daughter cells. It only had one copy of chromosome 1. Okay, remember that was in this type of structure with the centromere in the middle. And that's going to be pulled apart. Remember, in meiosis 2, we are going to be pulling apart sister chromatids. So we're doing a split like this. Okay, we're splitting it in half. And so in meiosis 2, this is going to go along just fine. One copy of chromosome 1 was there, and it's going to pull apart the sister chromatids so that now we will form 2 daughter cells, each with one copy of chromosome 1. Okay, they missed out on chromosome 2. It can't appear down the bottom. There can't be any chromosome 2 here because there was no chromosome 2 in this parent cell. Okay, I should just do that. So now if we look at the cell on the left, which is another abnormal cell, okay, in this case both of these 2 daughter cells are abnormal. Both of these are not what we want. So anyway, we have these 3 chromosomes, one copy of chromosome 1 and 2 copies of chromosome 2. During metaphase, I'll just draw over here, they will line up like this: chromosome 1, chromosome 2, chromosome 2. Okay, this cell doesn't know that it has 2 copies of chromosome 2. It's just going to treat it like any other chromosome. So when it divides, this division is going to happen properly, okay, pulling apart the sister chromatids, and so we'll have 2 daughter cells, each with one copy of chromosome 1, one copy of chromosome 2, and a second copy of chromosome 2. Okay, so in this case, when we refer to each of these daughter cells, you'll see we have this N number. Okay, remembering that N is the haploid number. Okay, so how many chromosomes are in a full set? Well, if you look at one of those cells, we had one copy of chromosome 1 and one copy of chromosome 2 is a full set. So how I got that is that in my parent cell here, it had 2 copies of chromosome 1, so 2C1, and 2 copies of chromosome 2. Remember, this cell must be diploid, because only diploid cells will undergo meiosis. And so half of that would be one copy of chromosome 1 and one copy of chromosome 2. So N, or the haploid number, should be 2. Okay, a normal gamete will have 2 chromosomes, and a normal somatic cell would have 4 chromosomes. So when we refer to these, you'll see we call this one N plus 1. Why? Because it's meant to have 2, instead it's got 3. So it's got N, that 2 plus 1. Same with this one over here, both these are N plus 1. And these were meant to have 2, but they've only got 1, so it's N minus 1. Okay, so with non-disjunction in meiosis 1, you should be able to draw this process down here, okay, actually, or the whole process, but also just state that you'll have 2 daughter cells at the end with N plus 1, these 2 down here, and 2 daughter cells with N minus 1 number of chromosomes over there. Okay, so if we look at non-disjunction in meiosis 2, first of all, this cell has undergone meiosis just fine. Okay, meiosis 1 has proceeded just fine. We have this cell over here, which is normal. Okay, it has half the number of chromosomes it had before it split. It had one copy of chromosome 1 and one copy of chromosome 2. That's a normal cell. But this cell on the left also started off with that. One copy of chromosome 1, one copy of chromosome 1, and it had one copy of chromosome 2. Remember, in meiosis 2, we want to be splitting apart sister chromatids. Okay, we want to draw a line down there. Just like this side is what's meant to happen. This will receive one copy of each chromosome. This will receive one copy of each chromosome. But in this case, something goes wrong, and instead of splitting up down here, this chromosome doesn't split. And so the cell on the left is going to get 2 copies of that second chromosome, where this cell will get none. So we have one cell with N plus 1 and one cell with N minus 1. But over on the right, seeing as this has happened just fine, we have its 2 daughter cells are just N. They are normal. So non-disjunction in meiosis 2 results in 2 normal gametes, one N plus 1 gamete and one N minus 1 gamete. Now keep in mind, this will usually only happen to one chromosome at a time. Okay? It is not, so in this case it happens to chromosome 2. In a human, it would be likely that all the other chromosomes divide just fine. This is quite a rare event. It does happen, but it's meant to be quite rare. And so you will only have this N plus 1, just that one extra chromosome. If non-disjunction for some reason happened in multiple chromosomes at one time, then each of those would add an additional 1 to that number. Okay, I won't give you an example of that. I won't ask you about it, but just be aware that this is usually happening to one chromosome at a time. Okay, so if you think about this is the gamete, let's say this gamete is going to meet up with a normal egg cell. Okay, let's say this is a sperm, these are both sperm, and they are each separately going to fertilize one egg. Okay, now remember, an egg is also a gamete, and if it was produced normally, the number of chromosomes in the egg should just be N. Right? In a human, that would have 23 chromosomes. And so if these two fuse, okay, let's draw a, remember, okay, this has got 23 chromosomes, or N, this has got N plus 1, so when I add them together, when we fuse their genetic material, okay, we are going to end up, I should not have done that, that was too complicated, we're going to end up with a zygote